Good afternoon, sir.
Good afternoon. Good afternoon to all.
Good afternoon. Sir. Good afternoon.
Yeah. Has he come?
Yeah, he had come 5 minutes back, sir. He'll be coming soon.
Okay. Okay.
Welcome, sir.
Good afternoon, Professor.
Welcome, Arun, sir.
Good afternoon. Unmute.
All right, thank you.
Yeah.
Hi, Dr. Looney, after a long time.
Yes. Yes.
Hello, everybody.
Yeah.
Dr. Fino helped me in the morning.
Yes.
Good afternoon,
Good morning.
Yeah.
Good afternoon, sir. Good afternoon.
Everybody. Good afternoon.
Sir, we have Dr. Naveen Mohan, who is our principal there, sir.
Wait,
Yeah.
okay, hello.
Good
Hello,
afternoon.
sir.
Good afternoon, sir.
You looks very old now. What happened?
Hey, I'm not old.
You should not be. You should not be.
You
You're
look
from down,
as
right?
young as you were. Only,
Yes.
you look as young as you were 20 years ago.
I still remember, we have our JC, that, fellowship programs, maybe
Yeah,
20 before. Remember?
yeah.
You came with Professor Guha and all. I think he's no more now.
Who is that
It was-
person?
Professor Guha, who came, for the first time, the fellowship program you organized at Jorhat.
Right.
Remember? Yeah. I think
Right,
maybe
right.
20 years before.
Maybe about 30 years or so.
Yeah, something like that.
Yeah. And you were the SBC convener
Yes,
at that time.
yes.
You forced me into a lecture which I was not
Right.
prepared for at all. Right?
Yeah.
Okay,
Anyways.
good day.
Yeah, I think we'll start now.
Do you go to Jorhat anymore now? You don't.
Yeah. I'm going. I used to go. Even January I was there this year.
Oh, really? Okay.
Because of the COVID, I'm not in a position.
Okay. But this is a good venture that you have taken.
That's what I thought.
Yeah.
Yes.
Okay, sir, let's start, sir.
Yeah, yes.
Okay, very good afternoon to all of you. Today, we are on to the 9th day of our e-conference, Current Trends in Life Sciences. And today, we are blessed to have Dr. Arun Lahiri Mazumdar, who is a INSA Senior Scientist, Division of Biology, Plant Biology, Bose Institute, Kolkata with us. I welcome him to this event. Welcome, sir.
Thank you. Thank you very much.
Next, I'd like to welcome Dr. B.G. Unni, who is our advisor as well as Director, Academics and Research to this event. Welcome, sir.
Thank you.
Next, I'd like to welcome Dr. Naveen Mohan, who is our principal as well as convener of this program to this event. Welcome, sir.
Thank you, Dr. Finos.
Saying this, I'd like to welcome Ms. Shinsi P., who is the head of the department, Biotechnology, to introduce the guest to the audience. And I also like to welcome all the audience to this program.
Okay, sir. Good afternoon to all. It's really an honor for me to introduce our today's guest speaker, Professor Arunaindranath Lahiri Majumdar, INSA Scientist, Division of Plant Biology, Bose Institute, Kolkata. He received PhD Botany from Bose Institute, Calcutta University, and DSc in Biochemistry from Calcutta University, 1985. He was formerly Raja Ramanna Fellow at Department of Atomic Energy and Senior Professor and Head of Division of Plant Biology and Acharya J.C. Bose Biotechnology Innovation Center, Bose Institute. He was a visiting fellow and associate at Fogarty International Center, National Institute of Health, USA. He's a lecturer, reader, professor and head in various universities in USA. He's a fellow of National Academy of Sciences, India, Indian National Science Academy, Indian Academy of Sciences, and West Bengal Academy of Science and Technology. His research areas include plant physiology and biochemistry, abiotic stress responses in plants, and plant biotechnology. He received several awards and honors like Career Award GC, Sir Rash Behari Ghosh Traveling Fellowship at Calcutta University, Overseas Associateship DBT, Sadagar Swami Endowment Award SBC, Foundation Day Award Bose Institute, Rockefeller Biotechnology Career Fellowship, member of editorial board, Plant Molecular Biology reporter, Parija Memorial Lecture Award, Utkal University, elected member of Guha Research Conference, elected vice president of Society for Plant Physiology and Biochemistry, S.P. Saxena Medal Award from Indian National Science Academy, S. Patnaik Memorial Lecture from Utkal University. Thank you all. Now I would like to invite Professor Arunendranath Lahiri Majumdar for his presentation.
Thank you very much. And I never expected that somebody will introduce me with this long words. I thought that Dr. Uni will introduce me. Arun, here is Arun, whom I know since last 30 years or so, and he missed my earlier invitation because of some family problem. But this time I could get hold of him. Isn't that Dr. Uni? Anyway, thank you very, very much, Dr. Sensi. And let me try to present my talk. And I need probably Dr. Finos's advice and try to follow you, sir.
Yeah, sir. Sir, you
Okay.
can go to the present now, sir. Present now. It will be on the beneath
Beneath.
on the right hand corner. Present now. Then your entire screen.
Wait a minute. Yes, I got it. Present now.
Entire screen.
Okay.
Then a window will open up and you can click on it and then
Here.
then share.
Okay.
Yes, sir. Then you can go to the PowerPoint presentation now, sir. Yeah, you can make it
Is
full
it
screen, sir. Yeah, it is coming, sir. You can make it full
coming?
screen.
Okay. Is this all right?
Yes, sir. Please. Yes, sir. Okay, it's fine, sir.
Okay. Am I audible to everybody?
Yes, sir. You're audible.
Perfectly fine. Perfectly fine.
So can I start now?
Sure, sir.
Okay. Firstly, let me put my appreciation for Dr. Uni and the team for inviting me for this talk. And as you'll see that I made a little extension of the topic that I initially presented before you, including salinity stress amelioration as an example. The reason is very clear to me that when I started thinking about abiotic stress in plants, I think it's impossible to give everything under the sky of environmental stress. So I picked up the salinity stress amelioration as an example only. And let me give you some example of the environmental extremes of planets, Venus, Earth, and Mars, in which Earth is in between with water, carbon dioxide, and ozone. Hydrothermal vent is geoactive sea, is also under environmental stresses. In deep sea, we see the same kind of environmental stresses, temperature low between 2 to 4 degrees, pressure very high, light non-existent. Hypersaline environment on the earth, lakes or in the sea, environmental stresses are like that. Intertidal coastal areas have also the stressed effect in which are affecting the plant life. In deserts, location is sparsely distributed on Earth's surface, for example, Sahara or Atacama deserts. And we have also deserts in our own country. Environmental stresses are temperature high and wide daily amplitude of this kind, dryness very high, constant lack of water, high solar radiation. Environments are shaped by human activities also there. Anywhere in the earth where human activity has induced major changes, that will be all. The stressors are temperature, light, high concentration of polluting chemicals, which are heavy metals. Now, deserts annuals evade drought and wildflowers after seasonal rainstorm look like this. Eucalyptus leaf hang vertically to present less surface area to sun, only to avoid the stressor. Drought stress corn with rolled and necrotic leaves are also there. And you can see these are the adaptations that you have. Now, unique adaptations are also found in plants like this one or this or this or this. This one is Xerophyta, this is Selaginella. And mangroves live in the typical and subtropical estuaries. And when they come up in the air, this is what they find. The pneumatophores come here. These are the different kinds of things that you have. This plant named Salicornia is inundated by tidal water in the Sundarbans. This is something which we collected and saw that in the Sundarbans near the Bay of Bengal. This is a wild rice, salt-loving rice, which is called Porteraceae, growing in the banks of the Sundarban area, which is also inundated by the tidal water when the saline settled water. They survive. This is another one in Salicornia. You see the dry, salty river banks in the upper. And it's so much so, you can see the salt here and also the drought causing the soil erosion here, or the breakage of the soil surface. So both drought and salt affect these plants, but these are surviving. Now, being basically sedentic in nature, plants have to evolve mechanisms through which they can evade the adverse conditions they are exposed to. The ones that I have shown earlier is the ones where they are natural plants and they have adapted themselves to that particular environment. Now, like this, xerophytes, which are presented to dry environments, they have the mechanism of drought evasion, an entire life cycle during brief rainfall. And the adaptations are thick cuticle, dense hairs, sunken stomata. Succulents is another one which you see. Mesquite trees, long taproot. Roots die back when dry. They minimize water loss at the expense of adequate carbon dioxide. Saline environment in the mangrove, among the mangroves, halophytes adapted to saline habitats. They transport sodium and usually chloride ions to leaves. Ions are stored in the central vacuoles. Salt glands exceed salt, and they're removed by rain or water. Now, similar adaptations we see in halophytes and xerophytes. Many accumulate proline, one amino acid, more negative osmotic pressure. Succulents is very common among them. They are reduced leaf area and thick cuticles, shallow roots, oxygen is limiting, oxygen is low, so roots use fermentation instead of the normal respiration. Pneumatophores grow out of water. Lenticels allow oxygen to diffuse through. These are the different kinds of adaptations with both halophytes and xerophytes take advantage of. One has to remember the xerophytes and halophytes, one thing is common, both are water deficit conditions. Halophytes because they have more salts and xerophytes because they have less water. Well, these are the natural adaptations of different classes of plants which inhabit various environments. What happens when plants growing under normal conditions are subjected to adverse conditions? This is the typical questions that we ask when we think about plants growing under adverse condition apart from the normal one. Do you learn anything by these halophytes, xerophytes, and the extremophiles? Do they have their own mechanism, the genetic makeup, which can be adapted to these plants, which can grow under abnormal conditions now? That is the very questions that we always ask. Now, how to change the mechanism? The environmental stress conditions, both abiotic, such as heat, salinity, decreased water availability or dehydration, or biotic, which are insect or pathogen attack, can have devastating impact of plant growth and productivity, potentially resulting in extensive loss to the agriculture. particularly for those crop plants on which we are dependent. And most of the crop plants, incidentally, are salt or dehydration sensitive. So it collapses the whole economy as well as the ecosystem. To withstand harsh environmental conditions, plants evolve sophisticated perception, signaling, and acclimatization mechanism that allow them to survive different stress conditions, even at the cost of reduced growth and yield. At times, they can survive under such conditions, but there are examples in which you have the growth and the yield are reduced. However, as one can see, the successful acclimation of plants to stress conditions require an efficient, timely, and coordinated response that spans most, if not all, parts and tissues of plants. Let me explain it through this slide, which is a little, I wouldn't say complicated, but it's a little difficult to understand at this stage. The example of dynamic response syndrome in plants are like this. You have a plant, and these are abiotic stress and photosynthesis, how they're affected, biotic responses, how they affect the different genes and others. Organ and plant growth is also affected badly. So in which a number of components in the plant biology and the plant system are taking parts, and they do act at a stage in which all the activities of the plant biology are covered. Now, to come to salinity is a major abiotic stress, and it presents multiple changes in the production of croplands. 77% of land surface and 5% of the cultivated land are affected by salinity. This has been long back worked out by Flowers et al., but it still holds good. And in fact, by virtue of the different fertilizers, this keep changing every year. The salinity condition reduces plants' ability for water absorption, resulting in reduction of growth rate, which negatively affects many metabolic changes and finally leads to cell death. It all starts with the plant seeds and their germination. This is a condition where it's a normal behavior of a seed when it starts germinating. Micropyle is a place where water gets into the plant seed, and by virtue of the gibberellic acid in ABA interaction, dormancy is relieved and amylase enzyme is synthesized, in which starch to sugar conversion is mediated. This sugar enriches the embryo and the embryo starts growing. Now, what happens in presence of sodium chloride in a saline condition? Salt water doesn't allow the micropyle to function. So there will not be any water inside in it to this. And this is the whole process is inhibited. So the synthesis of amylase as a result of GA-ABA interactions will not be there, and the dormancy will prevail. Now, because there is no amylase, starch and sugar conversion will be inhibited, and there will not be any sugar for the growing embryo. This is the first effect. Now, key impacts of salinity on a developing plant, it starts with the seed germination, water restriction, ion toxicity, ionic imbalance, photosynthesis reduction, biological nitrogen filtration reduction. So the overall effect of these in the growth reduction. What salinity affects also biological nitrogen fixation, I just mentioned. The rhizobium root, legume rhizobium consortium, the symbiosis is affected, in which this is the normal one. However, in presence of salt, the whole system collapses. And so there will not be any nitrogen nutrition to the plant in a condition when there is a legume rhizobium symbiosis. So certainly nitrogen fixation and nitrogen nutrition is affected in the plant. Now, how response is maintained in plants? The salt response is a multigenic trait in plants, and this is a quote from a number of workers in which they said it's not a single gene effect, it's a multigenic trait in plants. It can affect different types. One type is a membrane transport and uptake. Obviously, the uptake of water, uptake of solutes, and the regulation of this through the membrane transport is affected. These are the different acronyms of different genes which are affected. Antioxidants are also affected. One of the examples is ascorbic acid. Compatible solutes are synthesized in presence of salinity, which are polyols, polyamines, and amino acids like proline. These are also response to this. And the gene expression also affected and responded. Cellular protection is provided by LIA, one protein which is responsible during the desiccation of seeds. Ion homeostasis is another one. ATPase, calcium ATPase, and for general defense is PRP protein, beta-glucanase, and SODs. For regulation is phosphatidylinositol phospholipase C, MAPK and MAPKK are the two different types of things. Now, among the compatible solutes, these are synthesized by different types of pathways. And let me mention one thing to you, that these are pathways which come from different pathways in different plants. Not all plants can produce each and every one. The compatible solutes are also productants. Proline is synthesized by a mechanism which is available not in every plant. Glycine betaine is such, from the choline precursor, is not produced by all. There are families of proteins which can produce only proline or only glycine betaine. And cyclitols, which are inositol and pinitol type, this is a methylated inositol. However, this is one compound which is present in all organisms and required for their growth and development. Depletion of inositol leads to cell death, terms and inositol-less death. This is something which is very curious. I mean, this is present in all organisms and required for all growth and development. That means it is also synthesized. We argued several years ago that then it must be synthesized by all organisms, and the depletion of inositol, that means when this particular synthesis is blocked, it will lead to cell death, which is termed as inositol-less death, first shown in the Saccharomyces cerevisiae or Neurospora. And later it was also shown in animal systems, okay? Incidentally, inositol is present both in plant, animal, and bacterial system, all the biological systems. Now, if I look into the overview of inositol-derived intermediary compounds in plants, these are several inositol phosphates coming from inositol itself, which is synthesized from glucose 6-phosphate. Is my pointer working well in the presentation?
Yes, sir.
It's working.
Yes, sir.
Pointer is working, so okay.
Yes, sir.
Yeah, working.
Second type, this inositol polyphosphates and phosphate storage and signaling and growth. Phosphatidyl inositol derivatives, which are phospholipid derivatives, all these are also involved in signaling and membrane traffic, apoptosis, and epigenetic regulation. This is something which is very new these days. And this also produces a lot of compatible solutes in which inositol itself, pinitol, which is a methylated inositol, galactinol, raffinose family oligosaccharides, these are all derived from this precursor, which is involved in stress tolerance, signaling, sugar sensing, and storage. Cellular polysaccharides are also involved in the growth. So you can see that one particular molecule is involved in a number of biological phenomena which are related also to stress. How this is synthesized? There's only one single enzyme, which is called MIPS, encoded by inositol 1-phosphate synthase, encoded by INO1 gene, first discovered in East back in the 80s, early 80s. And of all the different types of inositol, this is the most, myo-inositol is the most prevalent, and these are all other epimers. Of the 9 different geometric isomers, this is the most biologically prevalent inositol which is found in the biological system. Others are available in different forms in different plants and other organisms. Now, when you started thinking back in the 80s and late 90s, late 80s and early 90s, we started thinking then if this is so, then let us first search for the protein and its gene in the diverse phyla. What you find that this enzyme is distributed across the spectrum of life. Underlying names indicate organisms with reported MMPs protein in the angiosperms, gymnosperms, pteridophytes, bryophytes, green algae, red algae, and chloroplast even, cyanobacteria, eubacteria, and archaea. And this in the whole in the fungi, protozoa, and in the animals, both invertebrate and vertebrates. Since almost everywhere you find this gene, find this protein, the first attempt for this cloning of the gene was successfully Saccharomyces cerevisiae in the early 80s, mid 80s. Till date, we have 172 INO1 gene sequences that have been requested for different organisms, including, of course, many that we have reported. The full-length gene sequences obtained from 48 prokaryotes, 25 archaea, and 49 eukaryotes. This 1-year-old data. It must have increased in the meantime. Now, bioinformatic analysis of all the MIPS sequences that you had, combined with the analysis of all available crystal structures. In the meantime, of course, there are 3 crystal structures that were available of the coded protein, showed beyond doubt that all MIPS share a common core catalytic structure. And several key amino acids are conserved throughout the evolutionary spectrum of MIPS sequences. I'm actually making a very long story short, in which we have shown that the bioinformatic analysis of all the sequences available of the 172 MIPS sequences, and we found the evolutionary relationship of the taxa for all the 72 planta, animalia, protista, fungi, between the prokaryotes and the eukaryotes. Obviously, the eukaryotes have a lot more sequences that we have obtained rather than the prokaryotes. However, when you took the total conserved nature of the MIPS enzyme, because of these things, and that we know that the synthesis of this particular compound, a compatible solute again, is mediated by one single enzyme, not by many, just one single enzyme. We started looking for the conserved nature of this throughout the evolution. And to cut the most, 7-8 years of work into one, I say that the catalytic domain is like this. And we found that it's only this pentapeptide, KEISK, between 355 to 359 is a representative in the West, that means rises at MIPS. These numbers refer to the sequences of rice sequences. But this is the invariant amino acid pentapeptide, which is prevalent everywhere. We concluded that evolutionary analysis identifies a conserved pentapeptide stretch containing the 2 essential lysine residues. Just to mention you that lysines are very important, the 2 lysines are extremely important for the catalysis of glucose 6-phosphate conversion to the inositol. The mechanism of that was been worked out by a number of biochemists and chemists back in the starting from 1970s, and it was confirmed that by this work that this is confirmed that why two licenses are so important. Okay, so from with these backgrounds, the focus of the laboratory, our laboratory, was this particular pathway. This is the pathway in which inositol goes into different compounds. And if you look into it, that this is the funneling reaction, glucose 6-phosphate to inositol through this enzyme, INO1 gene, and via the production of inositol 1-phosphate. This is the phosphatase which cleaves into mannitol. And this is a pathway in which the methylated inositols are produced by inositol methyltransferase. From now on, we started working on these two pathways, knowing fully well that this pathway is the one in which is an evolutionary conserved pathway and probably synthesize a lot of inositol. One more point I'd like to mention here, that this is the reaction which funnels this carbon skeleton to either this pathway, inositol phosphates, or to phosphoinositides, the membrane traffickers, or to these pathways in which the indolacetic acid, the plant growth hormone, and inositol complexes work, and or these sugars or carbohydrates, or these galactorylation pathways. However, so you see the tremendous power of this reaction, or the tremendous importance of this reaction to keep this whole metabolism going on. And in the event that there is a stress regulation of this particular pathway, there is every possibility that all these reactions will be affected, because the metabolic pathways will not be fed as it requires through this mannitol production. That's the concept that we started looking for. Most of the MIPS sequences that we have discovered or worked out so far was dehydration and salt sensitive. If you require all these things in a growth pattern of any animal or plant cell, how do you account for it? How do you assure that these are synthesized even under conditions of stress? And we know that if you have depletion of that inositol concentration in the plant system or any other system, be it plant, animal, or other cell system, depletion of this would lead to inositol-less death. So is there a plant source for such substrate regulatory inositol pathway genes? This is what we asked ourselves. We switched our attention to the wild rice, Botrytia quartata, which is also synonym of Oryza quartata, which grows abundant in the saline coastal belt. If you remember the first few slides which I scanned through, that adaptation of a particular plant in either in the desert or in the seashore, it gives you an idea that there will be, apart from anatomical and morphological adaptation, there will be biochemical and genetic adaptation of those plants to allow them to grow under conditions in which up to 500 millimolar sodium chloride can be in the soil, in the water, or everywhere. I mean, they will get into the plant to affect the physiological changes which is inherent of the plants. And we reason, because we are interested in the rice biology, we are looking for something which is very related to rice. And this is a rice, wild rice, which doesn't produce, which is not really the domestic rice. It's only used as a fodder. But we switched our attention to this particular plant. We worked out this halophyte as a halophytic model for rice. These are the things which are grown in the plant, either inundated by salt or under dry conditions. We brought these plants to our greenhouse and worked out and saw that these are salt glands in the lower surface of the leaves, and there are tears, salt tears, which accumulate salt. We analyzed them, and this whole seed, seed, we grew them under conditions of non-saline conditions, and we grew them into full plants. This is only our preparatory material for looking for the genes present in Portracia. Now, this was not unsuccessful for us. A couple of years later, we could isolate one salt-tolerant inositol synthase from this Portracia quartata. This is the Portracia quartata growing under conditions of the saline belt, and this is a normal Oryza sativa, our rice. But we could do that just by isolating the same enzyme from this. We reasoned if we have to have inositol metabolism going on in this plant, we have to have uninhibited synthesis of inositol by the inositol synthase under conditions where it will be protected. Fortunately, we came up with one enzyme which is salt tolerant itself. We isolated the genes both for the Portracia quartata. We named them PC-1 and along compared that with the OS-1, which is Oryza sativa-1. Both of them are for 512 and 510 amino acids, very close. And if you are very carefully look into this, the red part is the active part that I mentioned about, which contains the pentapeptide, which is required for synthesis for the catalytic activity. However, on analysis, we found that PC-1 and OS-1 are different for 37 amino acid stretch at this stage. They are different. Black and blue shows the difference between the two. And what happens here? This sequence is responsible for conferring salt tolerance to this protein. I'm making a very, very long story in a few seconds, that we discovered that this is a stretch which is responsible for salt tolerance of the protein. When you deleted that, the enzyme remained active. However, it's not sensitive. It's not salt tolerant anymore. You deplete this part, the red part, it will be inactive. So we confirmed that this is somehow responsible for conferring salt tolerance in the PC-1. And surprisingly, this salt tolerance domain came because of the slippage of one frame shift right here. OS-1 adenine is dropped in PC-1 here. So right here, there is a frame shift, which gives rise to 37 amino acids unless and until this adenine is placed back in PC-1. We had to do a lot more experiment to come to this conclusion that is simply a frame shift which gave rise to this salt tolerant domain. And when we worked out the molecular basis, we saw that 37 amino acid stretch is primarily responsible. This stretch may have arisen by frame shift, adenine deletion and reinsertion. A lot of biophysical work we had to do a lot of biophysical work to confer, which includes tryptophan fluorescence, BCNS, and modeling studies suggest influence of charged environment across this stretch significantly contributes to surface hydrophobicity, which probably helps screening out salt ions in maintaining protein-protein solvent interaction. Incidentally, MIPS is a tetrameric protein, so association of this tetramer is very important. Okay, so without going into much more details of this is what we conferred. that we have been able to get one MIPS protein coded by PC1 Potassium quartata-1 gene, which gives rise to a salt-tolerant MIPS protein. Now, can this gene confer salt tolerance to evolutionary diverse organisms, to any organism to start with, by uninhibited production of inositol under salt stress to find out our own, to confer our own or our own hypothesis? Well, we tried it with E. coli, yeast, Saccharomyces cerevisiae, rice, and also mustard. In all cases, we found that whenever there is a transformation of the system or the conversion, I mean, we have this particular gene introgressed in either in E. coli or in yeast or rice or mustard. This is a monocot, this is a dicot, this is a fungus, this is a bacterium. Everywhere there is an uninhibited production of inositol under salt stress, and all those organisms, transformed organisms, survived under salt stress. That gave us an idea that this is what might have happening, that we have been able to synthesize inositol under conditions of salt stress, where increased production of this one is possible by virtue of this particular enzyme, which is ubiquitous, which is present everywhere. However, if you take the PC1 gene, MIPS from the Potassium quartata, you'd be able to synthesize the inositol even in presence of salt. Okay. The second pathway that we
Vinod,
Sir,
what happened?
somebody has muted him, sir. Just a minute,
How
I'm
he
checking.
could? How? Is it possible?
Yeah, he has left, sir, actually. I'm checking on it, sir.
But he can unmute, na?
Yeah, he can unmute.
Yeah, please. Please unmute. Professor Majumdar, please unmute. Please unmute.
Arun, sir, please unmute your audio.
Hello, Professor Majumdar, please unmute. Hello. Akka.
Arun, sir, you can just press escape. You can press escape button, then you can go back to Google Meet and you can unmute yourself.
Yeah.
Yeah, you're okay now.
But sound, it's still mute.
Arun, sir, can you unmute yourself? You can go to Google Meet. You can unmute yourself.
Yeah, am I audible now?
Yeah.
Audible.
Okay. So
Only the slide has to come.
only the slide has to come.
Yeah.
You can click the PowerPoint again, sir.
Where did I stop?
Yeah.
Tell me.
Yeah, it's okay.
Yes,
No,
sir, you can do full screen, sir.
Okay, where did I stop? Tell me.
I will tell you. We let it go. Let it go.
Let
Ahead.
it go. This way.
Yeah. No, no,
Okay.
no. Just pass
Backward,
it.
sir.
Did you hear me up to this?
Yeah, yeah, yeah.
Yeah.
Yes.
From
Okay.
this, from this.
Then
Yeah.
the reason we started looking for this particular pathway is, am I audible now?
Yeah, yeah. Audible.
Yes,
Audible.
sir.
Okay, okay.
Audible.
So we looked for this pathway, the second pathway in which we would be able to convert this added inositol in presence of salt into a storage polyol in which there will not be further utilization. Incidentally, penitol is not utilized for any metabolic event. So anything that you produce here can go straight away. We wanted to see whether we can pull this pathway into a system in which stress tolerance can be produced. We in fact got the same inositol methyltransferase gene from the same halophytic wild rice Portretia quartata. So I've been able to show that increased production of pynetol and IMT transcript under stress is possible here. Incidentally, this gene is cloned from Portretia, but the IMT gene is absent in Oryza. It's absent in rice, but present in the wild rice. That means this is probably the mechanism by which inositol methyltransferase allows synthesis of pynetol only in case of the wild rice, the saline, salt-loving wild rice, but cannot do it in salt, in the usual domestic rice. So we thought that we'll take this gene out from Portretia quartata and push it into normal rice and to see whether the normal rice can now behave like the wild rice in this. Can PCE1 along with PCIMT affect enhanced salt stress amelioration? We tried that first in tobacco, the normal system which a plant biologist would do. And you'd see that when you combine this, one and IMT, this is what the transformed tobacco would go up to 400 millimolar salt growth. And this one also, one and IMT is go. Look the difference between the two. And here also, when it shifted to the chloroplast, this is the difference is right there. So we proved that probably by combination of these two, we can have a better salt tolerance in rice. Question is, will overproduction of inositol and pynetol in cultivated rice help in salt stress management? I put it in a big queue because we had a big queue there. We didn't know whether we'll be able to do it or not. I'll very quickly go through this transgenic rice production, which is a normal technology for any plant biotechnologist, in which we started towards production of transgenic indica lines with PC-INO1, first Portretia quartata INO1 gene, and Portretia quartata IMT, inositol methyltransferase gene, either singly or in combination. A thumb rule for checking any gene which you introduced in a system is whether they're allergen or not. Particularly when rice is a food crop, one has to be very careful. We tested this allergenicity test for this. One had no allergenicity, and the PC-IMT gene showed some match, which is much less the allergenic range of greater than 50%. It's a 25% match or 24% match. So we are safe that we can use both the genes for transforming a crop plant like rice. These are the cassettes, molecular cassettes for transformation. A very normal thing, very routine thing for this. These are the ACM analysis of the cali which are used for rice transformation. Very normal and routine thing for any plant biotechnologist who do it. You have to pick up the right kind of stage in which you can do the transformation work, rice transformation. And the strategy for obtaining the homozygous lines are like this. You collect the T1 seeds, self-pollinated plants after transformation and hygromycin selection, and then take the T1 plants take the T2 seeds and then make these plants which are phenotypically homozygous. This only can get, you can only get it under T3 plants. There's a lot of work involved in it, and you have to be very certain that you are doing it rightly. And many times we messed it. And we started with 255 or 309, and the DC means double transgenic, these lines, and ultimately ended up with one single line in which it was homozygous, and it's really, it follows the same growth pattern as the normal seeds. That was important. The best performing lines were isolated. I don't need to go into all this, but I would say that sodium concentration and the different types of the plants were analyzed. The summary is here. New leaves unfolded after 7 days. Normal growth observed 300 millimolar salt, except PCIMT, 100% recovery. Transcript analysis of 9 best performing lines were done, in which the INO1 transcript or IMT transcript or the double transcript was organized. Root growth was analyzed. You will see the 3 different lines actually gave you the right kind of root structure, which is important for salt stress analysis, because these are the organs through which water can circulate. Excuse me. The most efficient salt-tolerant transgenic rice lines were recovered this way. And the best recovered plants were, we named 3 different lines, 7PC, 10PC IMT, and 6DC1. These were actually analyzed through different organisms. In case of IR64, no plants recovered after 300 millimolar. Among the 9 different lines that we had, all the plants from 7PC, 1, 10PC, IMT, and 6DC lines recovered after 15 days of salt shock. We are able to make a comparative study of the transcript, protein, and the metabolite level under no salt and salt conditions. We expect increased inositol concentration and increased planetol condition on these conditions. So the transcript, protein, as well as the product, all 3 matched, showing that these genes, PCN1 gene in rice transgenic, actually increased production of the transcript, increased production of the protein. So there's no translational inhibition, and the protein actually produced, it was a functional protein because it produced increased product like inositol, or in another case, the pinetol, the IMT product. So all these experiments had to be done to prove that we are actually dealing with this. The agronomic characters, any person would know whether the agronomic characters are the same as the original recipient plant or not. The ultimate outcome was this. We got one salt-tolerant indica rice after almost, I must apologetically tell you this, it took almost 7 years of work in the laboratory. Both the genes PCN1 and IMT exhibited sufficient potential in planta for imparting salt tolerance, incorporated either singly or in combination. Synergistic effect of both genes in planta sometimes produce transgenic plants with compromised seed production. We explain that because it probably attributed to the overly channelized flow of inositol to form pinetol. Because when inositol is taken out for pinetol, you are depriving the system of some inositol. Agronomically, that's why this INO1 line had been found the best salt-tolerant line among us. The big question here, next, how does inositol impart salt tolerance then? attempting speculation in this line may be that the presumptive role of inositol, an essential component of cell survival against inositol-less death. It depends on maintaining a normal cellular growth and development under stress. That is something which is very important for a plant or system which grows under salt stress or under any kind of stress. How do you keep the normal cellular growth? For this, if you deplete the system of inositol itself, it will not grow. And let me tell you that under many conditions, this is the affected step. Inositol depletion is ensured under stress conditions. We picked up that particular portion, that particular reaction in which we can stop this by introducing a gene which can endure the salt stress even after in vitro up to 500 millimolar sodium chloride. So that was a strategy that we used. And secondly, there is a possibility that inositol actually acts as a switch to induce expression of stress tolerant genes in the system. The possibility was still there. So keeping the second option in mind, we did the transcriptome analysis of the best salinity tolerant line. Low, what we found there, this is again, I'm squeezing a lot of work in one particular slide and giving a summary of what we have been able to do. In this particular line, I'm only citing this particular line, which is the most successful rice line that you have as salt tolerant. 1756 transcript was upregulated and 1093 transcripts were downregulated. And surprisingly, in our hands, when you analyze all these transcripts in the macroarray system, 19 genes were upregulated in all comparative sets. These 19 genes are related to genes common for signaling response to stress. So what is the model that you have? If you can increase or if you can produce inositol under uninhibited conditions, under stress conditions, and be it dehydration, let me tell you, I'm showing you only the salt stress data. This is also true for dehydration stress and also probably under biotic stress. A lot more work has to be done by my juniors, by my students who are appointed here and there. But these 19 genes are actually switched on by a certain amount of inositol, which switches this whole set of, or the network of the inositol things. My students and postdocs who are involved in this work are here. Many of them are settled abroad. And also, here is one, Monoj Maji is at NIPGR. She is in Boston University, director of a bioinformatics center. This lady is in South Campus, posted. He is in Hyderabad. He's in Calcutta University faculty. She's in the faculty in the Calcutta College, a faculty in Calcutta University, abroad, settled in India, settled in Indian industry. All these students and postdocs were, he is a guy who is involved in Nagarjuna industry presently, the chief of the analytical section. All these persons are responsible for this work. I am only a spokesman. Okay. I would end with this normal Burlock's statement. Never think for a minute that we are going to build permanent peace in this world, an empty stomach and human misery. Thank you very, very much.
Thank you. Thank you very much. Thank you.
Okay,
you,
was
sir.
they audible up to the last? Now,
No,
Dr.
no,
Smith, tell me, how do I exit?
No,
Okay,
excellent, excellent.
Okay, how do I exit from this?
Oh, okay, sir.
slide?
You can just press the escape button.
Oh, escape button. Okay,
Yeah.
fine. That good escape. And then
Yeah.
how do I go back?
You can close it, sir. The presentation, you can close it.
Okay. Now I'm here.
Yeah, be
Yeah.
there.
Thank
Yeah.
you.
Now you can stop. Press the stop presenting button on your screen. On the left hand side, it will be
Yeah, good. Perfect.
yes, sir.
We could
Okay.
see him now. Now we could see
Okay.
you very well.
Okay,
I
sir.
can see myself. Man, I cannot see myself where I am.
You are there. We could see.
Anyway,
Maybe
where I am,
somewhere
I cannot see myself.
you will find some corner. You see. Check that. Click that. You
Oh,
can
okay,
get.
here, yeah.
Yeah.
Yeah, I saw it myself.
Fine, sir. Fine. Fine.
Okay, sir. Thank you,
Okay.
sir. The session was very informative. And I now like to welcome Ms. Shinsipi, who is the HOD of Department of Biotechnology, to lead the question and answer session.
Yeah, tell me
So
the
thank you, sir.
microphone.
Thank you, sir. It was a very informative session. And there are some questions for you. May I ask you?
Sure. If I can answer
Okay,
them.
sir. Okay, sir. First question is, what is the role of ascorbic acid in plant under stress? It's related to ascorbic acid. What is the role of ascorbic acid in plant under stress?
Okay. Ascorbic acid is also a vitamin C, and there is one extension of the pathway which actually comes from a galacturonic acid, which is also a pathway for minoxidil conservation. And it is a terminal metabolite in which the stress is affected. And it is used as good as any other osmolite or osmol protectant.
Okay, sir. Thank you. Another question is, does the high temperature affect activity of polyphenol oxidase in plants? Does the high temperature affect activity of polyphenol oxidase in plants?
Polyphenol oxidases are also involved in heat shock along with the heat shock proteins, and they do something with but it doesn't have anything to do with the dehydration part or the water deficit part. Let me add one more thing to the previous answer that I gave about ascorbic acid. Ascorbic acid is also serves as an antioxidant and is also responsible for SOS. I'm sorry, the ROS experience. Amelioration. Okay.
Okay, sir. Thank you. Another question is, what is the highest temperature plants can survive?
That's a difficult question for me. I
Okay.
don't know I really don't know how it can be, what should be the answer. I don't know the answer, but I would assume that it cannot go beyond 50.
Okay, sir. Thank you.
Unless there is a heat shock protein response. Dr. Muni would be able to answer that question. Muni, any idea about this?
I don't get much on that. Heat shock proteins, yes. What you said, that is the only point I have to say. But as far as temperature is concerned, how far it goes, and it's very difficult. Thermophilic, very difficult.
Xerophytes wouldn't take him anything beyond 50 degrees.
No.
That's my guess.
Yes.
It's very difficult to get something beyond 50, even for a xerophyte.
Correct.
With the sunken stomata and others. There are other faculty members. I mean, what do they think? Dr. Finos and Dr. Mohan. I think this is what it is. I cannot give you a right clear-cut temperature range where it can be done, but certainly it is never beyond 50. That's the maximum a plant would be able to ensure.
Yes, sir.
Okay, sir. One more question.
Sure.
Can a plant which produce fragrance resin responds to stress?
Please, beg your pardon?
Can the plant which produce fragrance resin responds to stress?
Well, fragrance resin. Is that
response to stress?
Fragrant rice. I know fragrant rice that it really, the aroma rice, so-called aroma rice, it really doesn't, it's not a salt tolerant rice. There is one example I can tell you what, that glycine betaine part, BAD enzyme, is also present in the aroma rice. And if there is a prevalence of the BAD enzyme, beta-aldehyde dehydrogenase enzyme, in those aroma rice, only those varieties can survive under conditions of salt or under stress conditions. Not all. So there has to have a correlation between the two. If there is in one QTL, if there is one QTL which will reside both the genes, then only it is possible. But that requires a genetic analysis of all the varieties, which are very different. I mean, not all rice varieties are also plant varieties will also do that. And if you're talking about anything beyond aroma in rice, that is difficult.
Okay, sir. Thank you. One more last question is there. Does leaf weight has an important role in screening for drought tolerance when compared to leaf area?
Leaf weight and leaf area.
Yes, sir.
Leaf, I would imagine the leaf would expand only when there is enough of water and the prevalent light incident onto it. And the weight will remain the same, but the leaf's size will be much different.
Yes.
Because they would try to reduce the surface area so that water loss can be minimized. That's physiological aspects of plants in which they try to put that. Remember the xerophytes, they do not have leaf structure. The spines are actually the leaves. The stems take the chlorophyll in them so that the photosynthesis goes on. But they are not actually the cladode and phylloclade, those structures, the morphological structures. The sunken stomata of these leaves are also another one in which they try to not lose much water in the plants. I don't know
Okay,
whether.
sir. Thank you. That's all about the questions, sir. Thank you a lot.
Okay, not much questions. I mean, these are only similar questions. I thought much more difficult question I expected. They're all biotechnologists all around. I know.
It's good for you. Thank you.
Right. Okay.
Yeah, thank you, Professor Majumdar. In fact, I have to say here, to get a person from Bose Institute, it's very difficult. And especially a person like Arun Lahiri Majumdar. It's not think that, I'm talking in front of him. I have to say, because my association with him, one of the topmost plant molecular biologists as on today. You can see that a very few persons has become, the very fellow of all the academies, Professor Majumdar. And his contribution, you could see that 7 years it took to have a salt tolerant, and that to the genes, and the way he explained. He was asking me that, how to go with the talk, because I know for a very hardcore molecular biologist to present the things in a very simple and lucid way, and that too some of the slides, you could see the way he presented, so simple. Even, 7 years work, he could present in 30, 40 minutes. Each and every faculties and the students, that make them, that's again a teaching. In fact, I could not bring him when we organized a conference a couple of months back. Now I'm very happy. Again and again, I have to express my thanks to Professor Arun Lahiri Majumdar for spending his time, and that too giving a wonderful talk. Each and every aspect is clear, and how to go for a salt tolerance strains, how to go for a genes, how much effort one has to put. Everything he has put it in a so loose style. It's an ideal example for all the faculties and the students to follow, and also how to go with the research to starting to get a product, and that too in a simple way. I again and again, thank you very much, Professor Majumdar, for spending your time and also giving a very, very informative lecture to all the students and the faculties in this part of the country. Certainly, we will be in touch with you. Thank you very much.
Okay, can I say something, that Professor Unit?
Yes.
Yes.
I'm thankful to you for bringing me back to your contact and your colleagues. And especially, I would thank Dr. Finos, who spent some hours this morning telling me how to do this. And even at the last moment, he was teaching me how to do this. I'm sure you have a very good colleague around you, and I'm very much impressed with their habit. And if there is a second call by you, I promise only that I would not say no. I would never say no. Okay. Thank you very much.
Thank you.
Thank you
Thank
very much.
you.
Thank you, Dr. Finos, Dr. Mohan, and Dr. Saini, please.
Thank you,
Thank
sir.
you all.
Next,
Thank you.
next, this session is open for vote of thanks. I'd like to welcome Ms. Shinsi P to deliver the vote of thanks.
Okay, sir. Respected chief guest, honorable dignitaries, teachers, and dear students, it's really an honor for me to say vote of thanks today on this occasion. First of all, I express my sincere and heartfelt thanks to Professor Arunaindranath Lahiri Majumdar, INSA Senior Scientist, Bose Institute, Kolkata, who gave a very informative session, an interesting session on how plants respond to environmental stress. I must mention our deep sense of gratitude towards you on behalf of Department of Biotechnology and Microbiology of GEMS College. Thank you, sir, for your great speech. Now, I'd like to thank our advisor and academic director, Dr. B.G. Unni, for his constant encouragement. Next, I'm also grateful towards our principal, Dr. Naveen Mohan, for his support. I also extend my thanks to all the coordinators, faculties, students, and other personalities for their active participation throughout this event. Thank you all.
Thank you, sir.
Thank you. Thank you. Thank you.
Thank you, sir. Thank you.
Thank you. And goodbye.
Thank
Bye.
you, sir. Thank you.
Uni, let us keep our contact. Okay.
In touch with you.
Thank you, Uni sir. Thank you, Naveen sir.